This is our boot camp. Number 2, dkNET informatics tools supporting the research lifecycle. And here we have Dr. Jeff Greth along with Dr. Koei Lin from UC San Diego and dkNET, sort of walking us through the bioinformatics tools that you need as a researcher and talking about sort of some tips and workarounds and what dkNET provides the researcher in terms of these types of tools. So welcome, Dr. Greth. And if anyone has any questions, we are recording this. But if you have any questions, please feel free to raise your hand or actually add a question in the chat, and we'll take them at the very end. Yeah, so let's start.
Okay, thank you, Monica. So I'm going to give an overview of informatics tools that dkNET is providing, really across the research lifecycle itself. So, make sure that I'm on the right thing. Okay, there we go. So first, since I know that these boot camps are spread out a bit, wanted to give a little bit an overview of dkNET, to let you know who we are. And so basically dkNET is a research resource information portal for biomedical researchers. And this is supported by the National Institute of Diabetes and Digestive and Kidney Diseases, NIDDK, at NIH. Really the goal, that NIDDK, gave to us was, to basically, as more informatics tools and more resources are available to researchers, to really try to create tools to assist researchers in using all the tools and resources that are available to them. So we have been running dkNET for a number of years, our lab is out of University of California, San Diego, at the Fair Data Informatics Lab. And there are really 5 areas of the portal that I will go through. Some of these there are some further boot camps that will go into into these in much more depth, and I'll point you to those as well. But this is everything from informatics tools to evaluate research resources that you have available to you. Fair data services where there was already a boot camp. Our first boot camp was around fair data. And so there are a number of tools there for you as well. The discovery portal is a place where you can get into a number of biomedical databases and in the hypothesis center. So I'll walk through these, but I think what, a better way to sort of view this is to sort of view that in the context of, a typical research life cycle. So, when one starts, looking at a question, you want to do some background research, find, other information that may be available to you, to start formulating a hypothesis. And so that's where, again, the discovery portal and the hypothesis center come in, where you construct a hypothesis. You then need to plan your experiments, ensure that you're using, the appropriate resources. You have to, meet certain NIH mandates for authentication. And then, you need to basically collect, analyze your data. Again, what tools are out there for you to do this analysis. Use the hypothesis center, to delve into additional information around some of the data that you might be collecting. And then when you publish your results, there are also requirements there for you. And so again, additional tools to support your citing, of your resources. And again, where you might want to place your data, since NIH mandates for sharing data, are going to become much more, are going to apply to more laboratories in 2023 with the new mandates. And so, all these services are available through, the dkNET page itself. One thing I did want to point out, about sort of dkNET itself as well, is that we also have available services and around bioinformatics tools and other available resources. We also have a webinar series. And so this webinar series we've been running for a number of years. All our webinars are up on YouTube so that you can view any of the past webinars. And then we're starting our new series this September, in late September, and you can check in and get information around a number of different bioinformatics tools and resources there as well. So, a lot of this is going to be driven by sort of a use case, just sort of give some context around, some of the questions that may come up. And so, we're going to walk through, just a simple use case around, diabetes and, its relationship to, problems with eyesight. And so, we'll walk through these various areas and sort of put that into context in terms of what the informatics tools provide. So, for example, just starting, that is a question of interest for you. First stop could be the Discovery Portal. And Discovery Portal actually provides a large range of information. So it's information about research resources, information and data that's available, but also has information on funded grants, so research that's already been funded, and also potential funding opportunities. So it provides a very large collection of information. So if you're searching within the Discovery Portal itself, you'll put in your search, and then you'll end up with a number of different category filters. And so these category filters allow you to focus in on what you're looking for. Again, because it's such a large amount of information, if you're looking specifically at information on funding, funded grants or funding opportunities, you can immediately narrow that down. If you're looking for data related to your search, so you're looking for existing data sets or data that you may be able to use in your research, you can immediately zero in on that data itself. The results themselves are then laid out in a list of all the online resources that might have that information available to you. And when you actually go into here, you'll click through to a page showing various results. And so within dkNET, we bring together both sort of DK specific resources, so there are a number of DK specific data resources, research resources themselves. And so, we bring those into the Discovery Portal, but we also have, resources that are more general to the biomedical domain that also, play a role, in DK. And so, we have more general resources as well as DK specific resources. So if we're, looking again at, diabetic eye disease, for example, we can also click into the literature, but also clinical trials. So there are a number of clinical trials resources, both in the US and Europe. And so, we provide sort of an integrated view across these. So, one of the important things to note is that there are many resources that potentially provide information about the same type of information. And so for some of these, we try to make it easier to review that by creating what we call these integrated views, where you can sort of search across clinical trials, be they from clinicaltrials.gov or from the European Clinical Trials Network and other sites. And so when you go into individual information, you'll notice that, each appropriate clinical trial has some basic information, but then also there are links provided, that will link out to that resource itself. So again, we go into many of these databases, and one thing to note is that we actually extract the data itself to actually provide this information. So if a site does not have a website that is easily crawled by Google or other crawlers, we're actually extracting the data from the back end. So sometimes, for some resources, that are more of a database than a website, we still have that information available, and make it easier to find sometimes than what might be available through the web, especially since Google does swamp you with with the literature mentions themselves for a lot of scientific queries and searches. So again, you may do some background research, using the discovery portal, but then, you potentially interested in some genes, that were either, outlined in some of the literature or, the clinical trials or other information from the discovery portal. And so, using SPP, you can start refining, that hypothesis. So again, the Hypothesis Center is something new in dkNET, which we're launching basically this, right now there's a soft launch on dkNET itself. There's going to be a full boot camp on September 10th around the Hypothesis Center, and we'll get into much more detail around the Hypothesis Center and SPP. What I wanted to do is just give a very brief overview here. And that, one of the issues with a lot of the bioinformatics tools being provided to the community is that, the barrier to entry for a number of these tools can be significant in terms of a bench scientist, being able to sit down and utilize the tools and also connect the tools. So how do you go from phenotype, to genotype to signaling pathways that you may be interested in? And so what we're doing in the Hypothesis Center is providing a platform, that allows bioinformatics tools to provide tutorials that will guide the bench researcher through using the bioinformatics tools themselves. And for a lot of bench scientists, we're also providing some more sort of scientific context around the tutorials themselves. So we're not just trying to say do step 1, do step 2, do step 3. We're trying to build in some context so that the researchers themselves get a better feeling, get a better idea of what the software is actually doing, when you're doing that type of analysis. And so, for example, within signaling pathways, if you do look at information around the consensomes and look at information around, the insulin receptor family, for example, if that's something that you're interested in, you'll get, a list of, highly ranked genes, that are involved in those pathways. And so, using this information, you can use this to, further tailor, your experiments, can find additional genes of interest that you may not, have been aware of before. I mean, again, Dr. Neil McKenna from Baylor College of Medicine will get into this in much more detail in his upcoming boot camp. And so again, once you've, worked through, the Hypothesis Center, now potentially have, additional, genes of interest that you're looking at, now you can start, planning, the experiments, looking at, what resources are available, and then also, making sure that you're abiding by NIH mandates for rigor and reproducibility. And so, the next informatics tools that I want to talk about are the resource reports. And again, in terms of resource reports and importance of authentication, much deeper dive is going to happen later in August. Again, what I'm going to do here is sort of give an overview, but also sort of give a walkthrough, of how you might use, some of these tools. And so, the resource reports themselves provide, overview of a lot of resources that may be important, for your research, for everything from antibodies, cell lines, biomedical software tools, protocols that people have published openly. So all this is available to you through these resource reports. And the reports themselves give a number, different areas of information, from the overview information that we can collect on usage and citation in the open access literature, that we can do natural language processing on in PubMed Central, and also providing ratings and alerts. So basically, again, using services similar to the discovery services, we can aggregate information about these resources themselves. And so, if we're looking at the PMAIP1 antibodies, some that we found through SPP, you can actually do that search in for example, the antibody resource reports, get a list of antibodies that are available. The antibody information itself will point you to the vendors, so you can go then to vendors looking for the antibodies. But again, the informatics behind this really is focused on identifiers for each of these resources. So each of these resources is uniquely identified through an RID, which allows us to aggregate information from many different resources. And then RIDs, if people aren't familiar with RIDs, and again, we'll go into much more detail, in a future bootcamp, but the RIDs themselves are persistent identifiers for research resources, and journals are, suggesting or requiring that authors provide these RIDs for resources used in manuscripts that are to be published. And they're really meant to answer simple questions like, to uniquely identify what resources were used in a study, something that was difficult to do in the past, in the sense of in terms of what information was being collected on in journal papers, journal articles. The information used to cite antibody cell lines was not always adequate, to identify the exact resource that was used. And then also you can start tracking, publications that were used by that resource. And then just some examples of sort of the resource ratings and alerts we get. So from, there are a number of different databases, for example, that have information about resources. So one of them is the Cell Source database, which is maintained by the International Cell Line Authentication Committee. And they provide information on, for example, misidentified, contaminated cell lines. And so we can extract that information and then provide that to you. There's also, information about, how antibodies perform. And so we can also provide that type of information as well. And so, there, again, if you're looking at any of these resources and you're doing searches in here, you can actually start filtering the information by, for example, how many times we have found in this open access literature, things have been mentioned. So you can start sorting things based on their use in the literature, which may help you more easily discover, an appropriate antibody. And so you'll see that, there are the filters on the left. If you go into the table view here, in the prior examples, we were in what we call sort of the snippet view, sort of looks like a Google search result, but then you can also put this into a table, which sometimes helps, with sorting and filtering, to sort of whittle down the antibodies resources that you'd want to review. Same thing with cell lines. And here, one thing that again that we can add, because we go into the databases on the back end, we can actually look, specifically for cell lines, that are from humans, and again, being able to filter and sort, then bringing that into the report, where you can see more details, but then also go and find out where that cell line is from. And so again, these resource reports provide the ability for you to dig into resources across a number of vendors, right? So again, cell lines, antibodies, come from many different vendors, and so what we're trying to do is provide a way to sort of help you get started, in finding the appropriate resources and reagents. When you're submitting proposals around your new study, one of the things that you'll have to deal with are authentication reports, and really this is tied into NIH's Rigor and reproducibility guidelines. So NIH is really, pushing, for researchers to follow guidelines to ensure that, their research, is rigorous and is more reproducible. And that's also, I mentioned the research resource identifiers earlier. That's another sort of tool that journals are using, again, to make the research more reproducible, and that what exact reagents were used by the researchers. And so again, using some additional tools that the dkNET provides, you can add, for example, the cell lines and the antibodies, to a authentication report. And what this does is it basically creates a collection, and it updates. So whenever you visit, this report, if we found any new alerts or any additional information, that information will show up on this report. And then we link you to and provide information on, for example, validation methods to assist in your generation of any reports for the NIH. And so this is, again, something, very easy to do in terms of the tools that are provided. So again, you can search, within the reports themselves and add the information, or you can do it from the web page for a resource itself. Okay, so once you've planned your experiment, the next step is to actually collect and analyze your data. One of the things that we do provide is, a registry of software tools. There are many different ways, to get into this information. So if you're looking for image analysis software, you can search, again, the resources and the tools available to you. If you're looking at sort of the most used image analysis softwares, the top two are MATLAB and ImageJ, which is what you'd expect. Those are, the top two tools really used for image analysis. And that will provide, again, starting point to find information. For example, if you're now looking more explicitly at protein image, additional tools come up. So for example, the Metamorph analysis software, again, used by a lot of groups. What's interesting is once you go into the resource reports and look at the usage. So once we've, when we do, the scanning, the NLP, to actually extract information, from the literature, to show where, these resources are used. We can also start providing information on, associated tools that may be used. So again, it isn't surprising that, for, a microscopy system, and image analysis software, that you may also use ImageJ. Again, so if you're looking at various tools, you can also see additional, tools that may be available, that may also provide, that similar type of information. But digging further into, the mentions themselves, for certain tools or resources, one of the, useful things that you may have at your institution are other researchers who are using tools in similar ways. For example, if you have antibodies, here I'm sticking with the analysis software. So for example, knowing other researchers at your institution that are using the same type of microscopy control system can be very useful, when you're, trying to either, if you're interested in starting up, your own new experiments, you're setting up new equipment, but also if you're troubleshooting, to see if, anyone, has, additional helps that they could potentially provide. And so, the mentions themselves, we also extract the location information from from PubMed, and then do some geolocation enhancement to the records of the mentions, and so from for example, these are researchers in San Diego, since I'm at UC San Diego, who have used this software. I can find PubMed papers, that are related, again, to researchers that are close to me. But then, once you have found some of these PubMed papers, what we do is we provide an additional view on the original sort of PubMed abstract information, that we also provide information on all the tools, for example, that a paper has cited. So for example, different the antibodies used in the paper, in addition to the software tools, and model organisms that may have been used in the paper. Okay, and so walking through how you can use sort of the resource reports to sort of tease out tools that are available to you. Then it's also important, once you're done with your studies, you have your data, now you're ready to publish your results. There are other requirements now that come into play, and so this is really around the FAIR data resources, to assist you in meeting new NIH mandates. And again, there are sections on the dkNET site itself, that provides information on FAIR data resources, and provides both information on sort of research data management, and also on sharing of data. And so research data management is something that's often overlooked in training, and is something that's very important as new requirements for data sharing, are being released by NIH. That means you need to make your data available to an external party, to make that data available. And so if data is not managed properly, once you get to the end of the study, you're going to have a lot more work to do, to get your data in shape, to meet, the requirements. And so, again, there was a prior boot camp that focused more on the FAIR data, and then also within dkNET, we do have a summer program on data science, and we are, from this year's course, which just concluded, we're going to be making some of our lectures available, and there are 2 lectures that focus on FAIR data and data management that may also be of interest. The research data management also ties into the information you're going to need to provide to NIH around the data management and also the data sharing that you plan for your research study. So again, the NIH has released a new data sharing policy. So this is their final data sharing policy that they've been working on for a number of years, and it goes into effect in 2023. And again, this will apply to all researchers. Before, NIH data sharing mandates really only applied to specific disease fields, for example, autism being one of them, traumatic brain injury, that required sharing of information as, or to particular data types. So for example, sharing genomics data. So there's been a genomics sort of data sharing policy at NIH, and then also for large studies. So anything over $500,000 direct costs per year, also had data sharing requirements. But with the new 2023 mandate, this is going to apply to everyone. So all research funded by NIH, and all data that's being collected. So not just for specific data types, but basically for anything, that was used in the research itself. And one of the important aspects of developing the data sharing policy is, trying to tease out, where potentially, you could deposit the data. So is there a repository that is related to the work I'm doing, and can I provide, can they take the data that I have so that they take care of all the requirements for ensuring that the data is published correctly. And so again, we provide some tools around finding appropriate repositories, and we're working on additional tools that we'll release later this year in terms of trying to walk researchers through that process itself. And so this the listings we maintain is both DK specific repositories and NIH sort of wide repositories. So there are some sort of NIDDK specific repositories in terms of the output from for example, clinical trials being run by NIDDK. So the NIDDK has a central repository for things like that, but then there are also more general databases, data repositories that are available to you. And so that basically covers sort of the overview of the informatics tools within dkNET. I mean, again, I wanted to provide sort of a context for these tools that sort of outline how they sort of try to guide and assist researchers through their research process. And for a number of these tools, we'll have, especially on the Hypothesis Center and the Signaling Pathways Project, those informatics tools will have a much deeper dive. In the September Bootcamp, where Dr. McKenna is going to walk you through, many of the, analysis methods that you can use there. So I'll leave that open for any questions.
Yeah, this has been really great. Thank you so much for this sort of guide and, sort of an overview, but also some really granular, instruction on how to use these tools. I would really like to open it up to the chat. If someone can add a question, that would be fantastic. And I guess we have some attendees. Any questions at all? I guess maybe if people are not forthcoming with questions, let's ask, what are some of the biggest questions you face, Dr. Greth, when you have novices trying to first use the dkNET portal? So for instance, when you hold the boot camp in the summer for the undergraduate students, what kind of questions are they asking?
So I mean, I think for a number of the students, especially in the discovery parts of the portal within dkNET, the amount of information that is actually out there can be quite overwhelming in terms of the number of different data resources that may have information about a gene of interest or a disease or phenotype that you're interested in. And so learning or figuring out how to best sort of navigate through the categories and sort of the filters that are available to you is useful there, just because of the large amounts of data that might be returned. The other thing is, finding or knowing, that tools are available, for certain, for certain aspects of the research life cycle, to support the researchers. So I think that's another, the resource reports themselves, provide information for citing the RIDs, so the research resource identifiers when you're publishing, but they actually as we walked through them earlier, they actually provide a lot more information. So being able to use that information to its fullest extent, in terms of looking for or finding resources that are available to you.
Yeah, that's good. I mean, that's a good appraisal. I also sort of, let's go to the other end of the spectrum. When when you interface with more expert users of the portal, what kinds of what tools are they sort of using? What how are they using the dkNET portal?
So I mean, there are different classes of expert users. I mean, in terms of informatics tools, I would say that the getting into some of the analyses, for example, through the Signaling Pathways Project, is really one area where expert users may go, and that really again allows you to take, for example, the signaling pathways, genes of interest that you're interested in, and really tries to pull out additional information, that could augment, what you have originally targeted, right? So, using, data that is freely available. So again, there are different levels of expert users, right? So expert users who, are really bioinformatics savvy, could potentially go to, geo and, do the additional curation that's needed, to extract data and do some of this analysis themselves. But what we're trying to do is provide sort of the that curation and provide a layer on top of that 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 you don't have to be a bioinformaticist who needs to analyze this by themselves, but by providing a tool that you can start exploring the data that is already publicly available.
Yeah, that's great to sort of appreciate that really really three types of researchers can really benefit from the use of the dkNET portal. So, and we'll see this as we, engage in the D challenge, where we're inviting novice users, intermediate users, and expert users of bioinformatics and other tools to use the dkNET and associated SPP or signaling pathways project to mine a curated consensome and come up with new hypotheses. And it's a very exciting times. I think dkNET has so much to offer, and it's a great, it's just a fabulous resource. Here's a question that just came up. While searching the resource of my interest using dkNET, there's so many resources in the results. How do I decide which resources are best for my experiment or data analyses? Can you answer that?
Yeah, again, I mean, I touched on it, when we were walking through the reports, but there are a number of different ways to sort of provide additional information or context around the resources, that you may find. And so one of them that we walked through was sort of the number of times we're able to find mentions of the resource in the open access literature. So that's sort of one marker of utilization by the community. I also pointed to the ability, for us to provide additional information, either characterization information on antibodies, information on cell lines, provided by authorities, if there are any issues, for example, contaminated cell lines, that may be out there. And then also, being able to sort of discover, researchers locally, that may be using similar tools that you could also talk with. So, I mean, again, there are multiple ways that you can provide that we can, where we're pulling in additional information for you to walk through that process, to be able to better characterize, the resources, that you are, finding in the search.
Yeah, that's good. That's a great answer. I want, okay, last chance for any questions from the audience. All right. Well, I think this was a really informative, boot camp. Appreciate you taking the time to share with us, and we will be hosting this on our YouTube channel for those who couldn't join us right now, but they'd like to learn more about the tools, the bioinformatics access that we have available through dkNET. So thank you again, Dr. Greth, and we look forward to the next one.